Good evening, good night, good afternoon, good morning, wherever you are. Today I am with you with another talk regarding functional genomics. Let me start. This is actually, as you see, 48 slide PowerPoint presentation. This was also a homework that I did in fall of 2006 in the class for functional genomics. So this will be better, I guess. So I'm just gonna do it as a presentation. Functional genomics, and let me start with this. If you are into medical biology or any molecular biology career, this is the course to start with, functional genomics, because now we are living in the space age, and at this time we don't know where life has come from, so that is the starting point. So we have this connection to space, the unknown, all these proteins and the RNA, DNA, where did they come from? We're still trying to solve this puzzle. Many unknown DNA sequences, many unknown, proteins, and in this vast ocean of bioinformation. That is the keyword here. Bioinformation, bioinformatics. So this is actually a long talk, but I'll try to keep it simple. Maybe it'll go over 60 minutes, maybe it's just 1 hour, 20 minutes or so. I'll try to make it short as much as possible. But it is to understand, why we are doing all this, because today we have first a Turkish astronaut just splashed down in the ocean, and a lot of people still have a hard time understanding why we have spent money on an astronaut that just flew into the space and did couple flips and this and that, and of course people are hungry, and there are many injustices going around, but it is about the new frontier. So that's the keyword, frontier. Because let me tell you this: while you are busy destroying this planet, while you are busy consuming this planet, a lot of people are busy trying to find new places to live. Correct? Am I right? I think so. Let me repeat. While you are busy destroying this planet, while you are busy consuming this planet, some people need to work, to generate new food sources, new places to live. It's just plain simple. Is that understood? I think so. So that's why it is called new frontier. The space is the new frontier, because maybe life has come from space. Maybe we cannot deny it at this stage. So with all said, let me start. So this was a sequence, protein sequence. All these letters, they respond to amino acids. Like methionine, serine, arginine, etc. See, it has the word salam here. No, no, it's not a protein belonging to a sausage. Okay, so this is a as you see, how many amino acids can you see? Just plenty. I mean, this is like maybe the three, then, six. So this is 10, and then you do the math. This this is like 10. This is actually a long protein. This is a long protein. Lots of amino acids, and what does it mean? Okay, good luck. Like it is like deciphering the Phaistos disk. They have found with the with the linear A on it. They're trying to decipher it. Okay, good luck. The deciphering life, deciphering hieroglyphs of the Egyptian history. Maybe it is easier. Deciphering Hittite tablets or Sumerian tablets. Maybe it is easier. What about deciphering life? Is that so? So this was the homework. The professor gave us this tablet, and then okay, just that, and told us that okay, find find what it is. So that's why I mean after all, he has taught. Of course, this was a homework to finish for us, functional genomics. And then imagine, like, this is 800 amino acids, for example, 800 times 3 makes almost 2,400 bases of nucleotide bases that codes this protein. So what is it? How can we understand? Imagine you have found a piece of rock that came from space, and then you have analyzed it. Okay, you found that the DNA in it, and then the DNA had this protein coding region that looked like this. What can you do? What can you do? How can you tell? Is it something from Earth? Or is it something from space? What the heck is this? So I think I think it's understood. Okay, we need a Rosetta Stone. That is to understand. I mean, here whatever I'm going to be showing here is not new. I mean the Egyptians already found it. They invented the Rosetta Stone. I mean, they translated some of the hieroglyphs into Greek language and other alphabets and etc. So what is this? Let's start. What can we do? What can you do sometimes? Okay, so first thing, you just have that protein sequence, which is full of amino acids, and you blast it. There's this page on the NCBI protein database. It's a search engine, but full of good stuff. I mean, it's a search engine that doesn't give you the results based on who paid the most money, which is a good thing. Thank God, that there is at least one search engine on this planet, that gives you the true, hits, I should say. So this is NCBI, National Center for Bioinformatic Information, Biology Information, or something in that line. It's bioinformatics. Okay, so that is the keyword, bioinformatics. Blast meaning comparison. You just take a sequence of amino acids, or DNA sequence. It doesn't matter, and then you enter it to the box provided on the site, and then press enter, and then the program, the search engine, compares that sequence to the known existing sequences in the database, and if it comes up with a match, it just lists, according to the similarity ratio. So it gives you this e value, and then some scores like depending on the quality of the match. So my first sequence is producing significant alignments. It's a matter of alignment. So it says putative conserved domains, meaning parts of protein, have been detected. Click on the image below for detailed results. That is on the on the actual page. They delete those blast results after some time, but they keep for some time, for you to save and etc. So it says pyroquinone, for the first result. Second, glucose dehydrogenase. Second, similar. Again, quinone, some protein related to bacteria. Again, the fourth one is glucose dehydrogenase. See, you had that strain, you had that sequence, and then it gives you all these similar proteins. That's how you start to understand, what is it good for. Next one. So NCBI blast first hit. So that means the first one. So I clicked on the first one, I guess. So protein, similar protein. Organism. It is from an organism, Serratia proteomerculans. It's so the sequence is like pyroquinone, similar sequence. So signal predicted, signal peptide, meaning if a protein has a signal peptide, that it is more likely to be located in the membrane. It shows on the report it has a signal peptide or not. See, between first amino acid and the 32nd amino acid, there is a signal peptide. So when the ribosome is synthesizing this protein, it just if you see in the first region, kind of the N-terminal domain of the protein, you see the signal peptides, and if there is any signal peptide, it is more likely to be located on the cell membrane. Calculated molecular weight. See, this is the molecular weight region. Region name GCD. See, glucose dehydrogenase. So there is a function. This protein seems to be doing a function. According to the first, comparison report, first hit. This is just the first. It's not saying 100% it is, but it just gives you an idea. So you make the decision. This is the first result. It's like I said, 800 amino acids total. Coding region, CDS region, coding region, total 803 amino acids. These are all these numbers, identification numbers. See, it could be a quinoprotein, pyroquinoline, quinone. It's just all those, quinone, the membrane proteins. So let's continue. Like I said, this is going to be a long one. I'll try to keep it as short as possible. My search started with NCBI NR protein blast, and my first hit aligned this sequence to a pyroquinoline quinone protein enzyme with very good scores. Corresponding link of first hit refers to a PQQ dehydrogenase. PQQ, that's the initials, PQQ dehydrogenase domain from a bacterial genome, which is 803 amino acid long. Other significant hits displayed alignments to glucose dehydrogenase domains with lower scores. Then I performed position-specific iterated blasts for the query sequence, and my results displayed similar alignments which refer to the same pyroquinoline quinone dehydrogenase domain with better scores than regular blast at NCBI. So you can tune down the blast, search to see if you can get a better match. Let's continue. NCBI PCI blast iteration 1. Like I said, okay, so sequences with E value better than threshold. Again, the first one looks like pyroquinoline quinone. First on the list. So seems like we are getting somewhere. Okay, let's continue. So second iteration, score bits. See, this says if you look at the scores, E value is 0. So the smaller the better. The smaller the better. The E value. The score bits 810. Let's quickly go up. The scores, see in this one 479, the score, and then E value is like that, and then go up. The first score is for 480. See, just if you tune the blast, then you get better scores. So again, it turns out to be just PQQ protein, pyroquinoline quinone. So this is another website outside this is another search, the Interpro results. So length, see the NCBI the first result was around 800 amino acids, because you have to match the length. That's a problem, that's just because if the query is query coverage, it gives you that ratio too. It has to cover all the amino acids. If you have like 1,000 amino acids and the results show match for like 800 or 900, it's still not enough. It could be still something else. You have to be able to find the function for all the amino acids. See, for 1,000 and 1,100 amino acids sequence. So first one that turned out to be flavotoxin. That's the second the ferredoxin. see the regions for iron and sulfur binding. So those are the spots. For example, the sequence has one binding spot for flavodoxin. So that's why it is called functional genomics. You're trying to understand what this sequence of amino acids do. Just basically. So you're looking at the different functions right now. What's happening within this protein sequence. So to PQQ, the pyroquinoline quinone. See, there are 5 different boxes, 5 different regions. Cytochrome C, cytochrome C connection here. Cytochrome C towards the C terminal. And in the middle section, there's a domain that looks like quinoprotein alcohol dehydrogenase like, in the see the towards the like N terminal, or the mid section. At the end, there's a domain. There's a section similar to cytochrome C, region. And then just at the beginning, remember there was the signal peptide at the beginning, the between 1 and the 38 amino acids. You expect to see the signal peptide if it is to be located in the membrane. And then see transmembrane regions. It's also looking at if this if this string of amino acids are located within the membrane, or crossing the membrane. That's called transmembrane regions. So let's continue. Interpro results. So this is like I said, another site. Apart from protein blast, it just gives you a number of overlapping proteins. See, they all look like each other. That's why life is more like 50 Shades of Gray. I mean, life is not black and white. Okay, so let's. It is to understand that first things first. Life is not a black and white phenomena. Forget it. There is no absolute black and there is no absolute white. It doesn't work like that. It doesn't work like that. It is more like 50 Shades of Gray. So let's understand. So this was the like search page, and then see that's the address. See, that's from UK. That's where this site is located. ebi.ac.uk. That's the database. Serine threonine protein kinase precursor. Endoplasmic reticulum to nucleus signaling includes serine threonine kinase. It is giving you. That's the address of the query. Hypothetical protein. So I think it showed to be the first one, methanol dehydrogenase subunit precursor. Methanol dehydrogenase. See, again, it's looks like a dehydrogenase protein, according to this website. And the second result is a hypothetical protein. So let's continue. Let's see if I have more. See, so what is PQQ? Pyrroline, quinoline, quinon. So according to the results, the first on the list is protein kinase. Second bacterial quinoprotein dehydrogenase. Third one pyrroloquinoline, quinon. See that matches 831 proteins total. That was the match. Wow, that's a long list. See, the professor gave us one sequence, and then it looks like 800 other proteins. They all look like one another. You know, it's not even 50 shades of gray. 800 shades of gray. You know, that's life. That's life. So what is PQQ? This PQQ, pyrroloquinoline quinon, is a redox coenzyme which serves as a cofactor for a number of enzymes, quinoproteins, and particularly for some bacterial dehydrogenases. A number of bacterial quinoproteins belong to this family. Okay, so it's a coenzyme. It's a redox coenzyme in bacteria. It's a coenzyme. Is it? According to results, according to the matches, just see, they already have, all this data bank. Let's continue. Overlapping interpro entries. See, overlapping proteins 79, 638. So this is 80% overlap. Average number of overlapping amino acids, numbers of overlapping proteins. And it says methanol dehydrogenase subunit precursor. First result, methanol dehydrogenase subunit. So more proteins, and this is bacterial quinoprotein dehydrogenase. That's the blue one. The green one. See, you see more green on the match, on the comparison. Pyrroloquinoline quinon. Quinoprotein alcohol dehydrogenase like. That's the red one. See, so that's the red one according to the result. And then see that these are yellow stripes and purple stripes and black stripes. Some extra information regarding domains. But the red one is kind of interesting. The red one. See, quinoprotein alcohol dehydrogenase like, and then there are other matches. Let's continue. So the summary of Interpro results. My search with interprotein blast aligned this sequence to several significant proteins, entry IPR 002372, including 1,2 quinon protein alcohol dehydrogenase like protein, and for bacterial quinoprotein dehydrogenase corresponding hit, referred to PQQ dehydrogenase repeat domain, which was also displayed in NCBI blast results with IPR cross-reference number. Results also displayed alignments to protein kinases and other smaller protein domains. See, it also looked like a protein kinase, but protein kinase is often found in the cytoplasm. And yes, there are membrane-bound kinases, but there were some alignments. So, quinoprotein alcohol dehydrogenases are a family of proteins found in methyllotrophic or autotrophic bacteria, meaning like utilizing methyl alcohol, methylotrophic, alcohol-consuming bacteria, or autotrophic bacteria. These quinoproteins use pyrroloquinoline, quinon as their prosthetic group. so it's part of it. There are three types of alcohol dehydrogenases. It's a big family of enzymes actually. Alcohol dehydrogenases. Type 1 includes methanol dehydrogenase. That's why you don't the moonshine guys no what is it like to drink methanol. It's a poison. It just makes you blind. It's just so, so if you're drinking moonshine, watch out, for methanol. Methanol dehydrogenase and ethanol dehydrogenase. Type 2 includes soluble quinohemoprotein with C-terminal containing HEMC. Type 3 includes quinoprotein alcohol dehydrogenase with a C-terminal cytochrome C domain. See, this is it kind of goes into the third class. See, quinoprotein alcohol dehydrogenase with a C-terminal cytochrome C domain. See, this is more like it, So that's why I made it bold, I think that says, okay, we have something here. These quinoproteins contain an 8-bladed beta propeller motif. See, these amino acid motifs in patterns, which is present in the N-terminal domain of quinoprotein alcohol dehydrogenase, ethanol dehydrogenase, and the heavy chain of methanol dehydrogenases. So they have something. It's a pattern. All these the search engines actually look for these patterns and motifs. It's all about these patterns and motifs, which life is full of. Okay, so among it's a family of alcohol dehydrogenases, and it just looks like the type 3 alcohol dehydrogenase. Let's continue. This is another site, FAM. Trusted matches, domains scoring higher than gathering threshold. 11 residues, 11 amino acids based on 11 amino acids, because it's a long protein sequence. I mean, it's just honestly, it's a long one. It's a big protein. So these are the scores. PQQ. See, it says domains are PQQ, cytochrome C. That was at the end. See, from 984 to 1062 towards the end. See, cytochrome C domain. And PQQ domain is more like at the beginning of the protein. See, around 549 and 83. These are the values, scores, that is assigned by the computer, the search engine. Another domain. So, see, that's the analysis of the the whole sequence. So some more results with some more values. Let's continue. Prefermentary PQQ accession number. That's the usually means identification number. Bacterial PQQ. PQQ enzyme repeat. The family represent a single repeat of beta propeller. See, that's the kind of the identification. This propeller has been found in several enzymes which utilize pyroquinoline quinone as a prosthetic group. So in the enzymes, if you find this propeller, then you can define better. So it's like a toolbox. Imagine somebody comes into your home or into your workshop with a toolbox, and by looking at the tools, you can kind of figure out who is that handyman doing. If he has a welding machine, you would say this is like a smith, ironsmith doing some iron work. If he has pipes and glues and the other things, and you would expect that, maybe a plumber, just like if it's a guy with a computer laptop and the electronic cables and equipment, and say it's a tech guy, so that's kind of figuring out, because that's the name, functional genomics. You're trying to understand what that enzyme is doing, and the proteins have toolbox. The enzymes have the toolbox inside. So this is to understand. Very important. This protein, it has the metal ions. It has the different domains and all these different propellers and the helixes and etc. The proteins have their own toolbox with them. So, this is a similar structure. See all these different parts, and and they show it, in the figure usually. If there's anything built inside the protein or inside the sequence. See, domains are listed here. Start residue 118, 155, 168. Because why? If it is built inside the membrane, then some parts of the protein are for anchoring the protein for physical purposes, and other ones are for chemical purposes. Think of a protein. There are regions for physical purposes like connection, binding, okay, and there are some domains that is required for the chemical work. For the chemical reaction to take place, you need amino acids, and to just if there's ATP to bind, and the substrate needs to bind. If this is an alcohol dehydrogenase, then they should have some amino acids reserved to bind in the cytoplasm, or outside the cell, or in the periplasm. The family represents a single repeat of beta propeller. The propeller has been found in several enzymes which utilize PQQ as a prosthetic group. This family forms interactions with other families. To view them, click here. See some extra information. And then it gives you information. Pyroquinone is a redox enzyme, coenzyme, which serves as a cofactor for a number of enzymes, and particularly for some bacterial dehydrogenases. A number of bacterial proteins belong to this family. Let's continue. FEM results. My query with FEM results detected several repeats of PQQ domain. Therefore, a connection to PQQ dependent enzyme family with entrusted values along with the cytochrome C domain. FEM results corresponds with HIT IPR Interpro results, the earlier search from Interpro, which refers to bacterial PQQ repeat domain. It also notes that these repeating sites are found in bacterial dehydrogenases, proteins. See, there's other name, bacterial dehydrogenase, or aka proteins. Bacterial dehydrogenases or proteins, which use PQQ as a cofactor. Let's continue. So I guess I am on the right path. Looks like we are getting somewhere. Okay, so another search engine, Civisprot results. See, every nation has their own informatics search engine. You know, because you have to spend money on this. You know, like I said, I'm just gonna repeat again. Should we invest in stuff like this? We should. Science is expensive. You have to store all this data. This is computer work. All the scientists around the globe are utilizing these search engines. And yeah, we are a proud nation, and we should do our own contribution to the bioinformatics. So this is I would like to remind again, as a nation here, as a member of the Turkish nation, we should do our part, you know, to this contribution. Bioinformatics, functional genomics. We should do our part. And for that, we need labs, we need equipment, we need biologists, and a law that helps biologists. We don't even have a law at this moment that just, you know, it helps with the employment of the biologist. That's the, I'm sorry, but that's the reality. We need better laws that help science and scientists. Okay, so Swiss prod results. Date run. See, that's the homework. See, 2006 and November, the 19th of November. See, that's the date. I did my homework. That's the date just before my the travel to Turkey to get my visa, because for this class, let me tell you this, I paid with my credit card this class, because at the time my TA-ship was not in place. It didn't start because I just I didn't get the student visa yet. So 2006. Okay, let's continue. Again, first hit with the significant score 462. E-value again, E. coli, quinoprotein glucose dehydrogenase. So kind of refining the search. Okay, so refining the search. Second score, quinoprotein glucose dehydrogenase. Third one, quinoprotein glucose dehydrogenase. Fourth, quinate schimate dehydrogenase. Fifth one, glycerol dehydrogenase, large subunit. So let's continue. Uniprot slash Swiss prot blast entry first hit. So that's the first, the first result. Entry name, DH, G, E. coli, primary accession number, this. So sequence was modified in 1997. See, it started. That's when they include that sequence in the database. First listed in 1990. Wow. Integrated into Swiss prot on 1990. See, the 1990s. They started building their bioinformatics, search engines. That's the start of internet, to be honest. That's the start of internet. So name and origin of the protein. Protein name, quinoprotein glucose dehydrogenase. So they have an extensive, search engine and an extensive database. So it helps with the definition and the diagnosis. Synonyms, glucose dehydrogenase, again, PQQ, same same. So name GCD. Taxonomy bacteria, protobacteria, gamma protobacteria, enterobacteriales, enterobacteriaceae, Escherichiaceae. Maybe it belongs to E. coli. So it's a better research. Nucleotide sequence, genomic DNA. So that's the amount of nucleotide bases. Cloning, mapping, and sequencing of the gene encoding E. coli encoding protein, the glucose dehydrogenase. So that's the publication. That's the name of publication. See, from this, I get based on this publication, based on this sequence in that publication. Because when you do a research and then if you get a sequence, you publish it and then put it in the database, and looks like from E. coli, E. coli protein glucose dehydrogenase. Let's see. Let's continue, and that is the score. 462. Let's continue. Uniprot Swiss prot results. My search with this database aligned this sequence to several significant proteins with high score assigned to entry P15877 protein glucose dehydrogenase. This enzyme is classified as PQQ also. Enzyme classification number of this entry is shown. So let's let me remind you that glucose is like an alcohol. It's not that you're gonna freak out, but if you look at the molecular structure of glucose, I mean, the stuff is full of hydroxyl groups. Okay, it just Google it. It just it doesn't act like alcohol, but it doesn't mean it doesn't have any alcohol groups. The hydroxyl groups. Sugars are like alcohols. I mean, molecular wise, they're just carbohydrates. It's just that's what they are. The same thing will happen. I mean, if you drink lots of beer, you will still get a belly. I mean, if you eat lots of bread, you still will get a belly bread. So beer bread, beer belly or a bread belly, same thing. So don't be confused with the glucose dehydrogenase. That's just the name of the enzyme. But if it works on those hydroxyl groups, and that's the function actually, dehydrogenase, and that means working on those hydroxyl groups and converting them to water or something else. So it's a detox enzyme to tell you this. Kind of dehydrogenases are often detox enzymes, work on alcohols or hydroxyl groups. So let me really quick. Let me check the time really quick. Oops, that is 48 minutes. Okay, 48 minutes. Wow, I just saw. I am on slide 19. Okay, let's just speed up a little bit. Slide 19. Let's move. Let's move. Okay, prosite signature pattern. Okay, so the signature means like special sections on proteins. Signature pattern. It speaks for itself. Okay. So let me just that's my search on the site called expasy.org, prosite. And that is the 4 hits in one sequence. So they're refined search engines, based on proteins. And see if there's any motives and etc. hits by profiles, cytochrome C family profiles. So we already knew that there was a cytochrome C domain at the end. So there's a total length, 1100 amino acids. So predicted features, so that found some motifs. See the sequence, SCL, RNG, the 1, 2, 3, 4. That's the score. The region, 982 and 1062. That's the location. And that is heme. That is the binding region for iron. Heme, covalent bonding. See, that's iron. See, metal binding features. So there's some other regions. So that's a flavodoxin. We already knew that from our first results. That's the signature pattern. See, the signature pattern search. See, this particular pattern is for binding to flavodoxin. See, it is named as flavodoxin signature. If you see this sequence of amino acids, that means it is for flavodoxin binding. And then the second for ferredoxin, for iron, for sulfur, iron sulfur binding region. See, that's the toolbox. Remember, every protein has a toolbox. Iron sulfur binding region signature. Again, see this see this area between the amino acids 344 and 55, almost 10 amino acid long. This signature. See, ferredoxin signature, and this is flavodoxin signature. And then it's a consensus pattern, as you see. See, that is the pattern, and the search engines look for these patterns for to predict the functions. See, again, consensus pattern, and see. So see, it's looking for these patterns, and then looking at these values. See, so there are 4 C's: 1C, 2C, 3C, and then it looks like 1, 2, 3, 4. Looks like a signature. This is important, and they did all these analysis for the COVID virus as well. I mean, remember, people still, the question whether the COVID virus came from a lab or not. And then for example, there was this furin cleavage site, which made it more virulent. People just still don't know where it came from. It looked like it was engineered. I am sorry, but a lot of people thought it was engineered. That it was because it looked weird. It didn't look similar to natural ones. That's the idea. If a sequence is natural, it looks like other ones, and if it doesn't, then you have to figure out where did it come from. Prosite signature pattern search. My search with prosite signature pattern search came up with 2 signatures, which were previously pointed by the interproscan earlier in the pages. One is flavodoxin, signature, and the other is ferredoxin, and that's iron sulfur binding region pattern. Okay, we found something else. Let's continue. Clustal W setup. So another blast. It says the name of the publication. Again, 1997. Protein. Uniprot, Swissprot, protein sequence database, search query. This is the length of the sequence. Remember, scores, the score 502, e-value, quinoprotein glucose dehydrogenase. See again, quinoprotein glucose dehydrogenase on this site, DDBJ, class W. Again, the second one is pyrroloquinone quinone precursor. The third one is glucose dehydrogenase, and so on. Let's continue. See, this is the spiral genes, NIG. You see Japan. See this search engine is from Japan. Spiral genes. The PCI blast. Okay, so. It's from Japan, and then first, see that's the score. Pyrroloquinone quinone precursor. Second one, quinoprotein glucose dehydrogenase. Third one, probable glucose dehydrogenase. Fourth one, pyrroloquinone quinone precursor. See, that is comparison. So you get the sequence. Analyze the sequence, and then if there is an coding region, turn that into a protein sequence, and then do a blast, and then you can figure out the function. Continue. My search with DDBJ aligned this sequence also several significant proteins with highest score assigned to entry Q1YG06. Quinoprotein glucose dehydrogenase. PCI blast, however, resulted in a different entry, which is PQQ cursor. Okay, let's continue. So our homework was about actually, doing all these, the entries in different search engines. Blast DP2 result, EMBL advanced blast. See, that's the EMBL site. Okay, so that's the date, I guess, 16 December. No, that's 1999. Washington University, St. Louis, Missouri, USA. Reference, blast query sequence. See, that's the name of the, that's the remember 1100 sequence amino acids query. And that's the blast. Searching. See, hold this that. It's just, even sometimes, because some of these servers are located in other continents, so you have to pick the correct timing. So I remember like professor saying, if it is overseas, you have to kind of do the timing, because if you on a regular work day, if everybody is busy and just loading the servers with all these queries and entries, and then sometimes it gets time consuming. This is actually some of these results. Computer takes a couple minutes to generate. It's not easy to compare all these sequences. So all these workhorses actually, these computers, so you have to respect them. You have to, and you choose the right timing when you're doing when you are doing the work. So high scoring segment pairs scores again. If it goes down, quinone protein glucose dehydrogenase, pyrroloquinone quinone dehydrogenase is similar results. EMBL advanced blast. Okay, so let's continue. EMBL advanced blast. Say between Swiss Institute of Bioinformatics and the EMBL outstation, the European Bioinformatics Institute. So I guess the European Bioinformatics Institute. There are no restrictions on its use by non-profit institutions as long as its content is in no way modified by this statement or is not removed. So this is for educational purposes anyway. So so that's the site EMBL, okay, and oxidoreductase, periplasmic, PQQ, signal transmembrane. See, those are the features it found. That's very important. And the first time, see, it mentions periplasmic. See, more detailed information. Oxidoreductase, PQQ, element. It has a signal peptide and it just has a transmembrane section. See, that's the location of the signal protein chain, the amino acid chain, quinoprotein glucose dehydrogenase, between 34 and 801. Transmembrane regions. See, now we're getting into detail. See, all these regions, transmembrane, meaning these amino acids in between 39 and 55, transmembrane, 59 and 79, built in the membrane, 94 to 108 in the membrane, 119 and 138 in the membrane, and then there is a proton acceptor potential, meaning. Let's continue. See EMBL advanced blast. See, more detailed advanced. EMBL advanced results. EMBL results were very similar to Interpro results with cross references. Query found references to PQQ repeat domains as well as quinoprotein glucose dehydrogenase. Results also supported that this might be a transmembrane protein with potential signal peptide presence. Data suggests that this protein might be a periplasmic oxidoreductase. See, the first time now I'm mentioning periplasm. Remember my earlier talk with the protoplasm and cell wall. So now we're talking about gram-negative bacteria, periplasm. See, we're getting somewhere. Periplasm, gram-negative bacteria, and then there were some other results with the mentioning E. coli, and E. coli is a gram-negative bacteria. Okay, but now it's talking about the location of the domains. Another blast, pure blast, glucose dehydrogenase again. The name of the bacteria, organism, Mesorhizobium lotte, Pseudomonas fluorescens, Pseudomonas potida, Agorobacterium tumefaciens, Rhodobacter spheroides. So glucose dehydrogenase, pyroquinoline, quinone, putative glucose dehydrogenase. These are the scores: 474, 473. These are the alignments, matching parts, overlapping, region ratio. So pretty much E values. Let's continue. So the first hit, okay. So the total sequence, glucose, glucose alcohol sigmate dehydrogenase validated. Bacteria, no, none from archaea. So oxidoreductase, signal peptide, periplasmic, metal binding, calcium binding, transmembrane, heme, direct protein sequencing, iron, respiratory chain. See, that's just see the keywords. See more detailed analysis here. Transmembrane protein, pyroquinoline, quinone, alcohol metabolism, NAD, the 3D structure, quinine, membrane protein, plasmid, So it helps you with the publications. And then the alignment tree, it's the phylogeny, domain architecture. So let's continue. Purple has displayed my protein as alcohol glucose cyclimate dehydrogenase. This protein sequence also displayed a bacteria protein in taxonomic range section. Okay, so it had the phylogeny option. Domain database, NCBI. See, domain database is different. See now amino acid database, nucleotide database. Domain meaning parts of the protein, large parts of the protein, that carry different functions. That's what we call the domain, like and terminal domain meaning the starting section of the protein. C terminal refers to the end of the protein, end section. So this is like this. This section is called end domain, like start. See, different sections in a protein are called domain. And again, this is a big protein, 1100 amino acids. That's a long chain. See, it's cytochrome C domain. See again, repeating domains. There's like 1, 2, 3. There's like so a long stretch of PQQ domain here in the middle. Again, another one towards the N terminal, another one towards the C terminal. All these repeats. See, we have these. Just why? Just now we can question. Why we have these repeats. Dehydrogenase, pyrroloquinolone, quinone is a cofactor. Because maybe due to the function of the enzyme. Like I said, in the cell, there are lots of hydroxyl groups, and you have to process them, and then you have to have enough protein, enough activity to cover all those, hydroxyl groups. So reductase enzyme, dehydrogenase. Let's continue. NCBI conserved domain database defined the query as repeats of PQQ dehydrogenase on N terminal and with a cytochrome C domain at the C terminal. Let's continue. TM pred modeling. Now we're getting into more detail. Possible transmembrane helices. Those spirals, because once you understand how amino acids are connected to each other, based on their connection, sometimes they make spirals, like when you bind them one after another, like making a wicker, the bag or something, it's like sewing, depending on the type, depending on the knot, they can get the different shapes. Women know this better. When they with the thread and with the needles and depending on the knots, you get a different motif. If you want spiral, then do this, and if you continue, like same pattern, same thing. So see these numbers from 10 to 29. And with these scores again, values. There's helices, and now we're analyzing the structure. Again, from 36 to 52, 65 to 83. So these are the regions inside the membrane, like transmembrane regions, 128 to 145, 20 to 6 to 144, and so on. Outside to inside helices, So this outside region. This is core center. See only significant transmembrane segments used. See so these are helices. Those are helices. N-terminus inside. See strongly preferred model. N-terminus inside. See the initial part of the protein is inside. And 9 strong transmembrane helices. So the N-terminal is inside the cytoplasm or periplasm. I should say inside. C1 429 from 429 see the values and the 39 to 57. You're like, this is now analysis amino acid by amino acid, all the way to the end, almost 1100. See, 1100 amino acids. It's not easy. It's a big protein. Alternative model. See different models. 8 strong transmembrane helices. Total score 12,000. See, this score is higher. See outside the inside helices. 9 helices. 11 helices. Let's continue. My template model query resulted in 2 strong models with similar structures. N-terminus was predicted inside the cell and the total number of 9 transmembrane. Maybe inside the cell or inside the periplasm. It's just this is see at the time. See, this was my first class in grad school. Maybe I mean like in the area maybe. So functional genomics. The periplasm, of course, periplasm is considered as part of the cellular structure, but I should be mentioning periplasm. Total number of 9 transmembrane helices were predicted for the preferred model with a total score. See, that was a good score. Transmembrane prediction servers. This is another search engine. Now we are predicting the transmembrane regions. Start, stop. See, start at 8, stop at 31st. Length the 24. Again start at 9 and stop at 19. Length 11. So those are the scores all the way to end. The transmembrane potential transmembrane segments. It just gives you the addresses of the segments. Results from this transmembrane database and corresponding length of potential transmembrane parts of the protein architecture. Total 20 membrane spanning segments were predicted with the cutoff values ranging from 1.7 to 2.2. Excuse me. Let's continue. Again, another site, top red. See all these different search engines found 10 segments, candidate membrane spanning segments. See, it started 4 first helix, started 4 the end 24. That's the score. Certainty, certain putative. There's some see, this one is maybe not as much. Lower score, certain putative, certain. So that's all the way to end. So it's the topology. See, it's just the geography of the cell membrane, of the protein, and the cell membrane. For top results were not too different when compared to previous predictions, with 10 transmembrane helices of similar orientations. Total of 4 structures were predicted with different probability values. Let's continue. See topology, cytoplasmic loop. See, it's more detailed. So it's cytoplasmic loop, transmembrane outside loop, transmembrane cytoplasmic loop, transmembrane external loop, transmembrane, etc., etc. So it gives you see cytoplasmic loop first. See the first amino acid. Stop at 3, and then see starts the transmembrane region at fourth amino acid and all the way to 24, and then it's so on. See this, see this chaos. It's just see this chaos. It's a long protein at 1100 amino acids, and every one of them is doing something. Every one of them is doing something. Isn't this crazy? That is the frontier. That is the frontier. That is the final frontier. This information, bioinformatics. Let's continue. MEMSET results. Okay, so MEMSET prediction results. See, a summary of topology analysis. First helix score, see the scores. That's 2 helices, 6 helices, and then go all the way to 20 helices. See that is the sequence. See I think those ones are the transmembrane regions. Those are X ones. See X's, the transmembrane regions. So let's continue. X central transmembrane helix segment, I inside helix cap, inside helix cap, inside loop, outside loop, possible N-terminal signal peptide. Let me go up. So that's the so this is see positive, negative. See outside loop, inside loop. Page down. See minus means outside loop, positive means inside loop. So positive means inside loop. These are inside the periplasm or cytoplasm. See these are outside loops. See these are outside regions. See what are these regions doing? See these are outside the membrane. These are exposed. These are exposed. Inside loop. The transmembrane. See these are it's kind of a physical attachment to the membrane. These are membrane proteins. Physically, it is attached, built into the membrane. See prediction. 1 for 13 to 30, 13 to 60, 63 to 82, and then so on. All the way to 1100. And let's say the C terminal is inside. Let's continue. MEMSET results. MEMSET result predicted is slightly less. 6 helices with significant scores. Prediction was based on MEMSET remodeling. It gives it a topography of query sequence predicting localization of helices, helix caps, and loops. Let's continue. Sort results, result information, plasma, see final results. Again, another search engine. Sort results. Plasma membrane, according to the results, it's a plasma membrane. The location. It could be on the Golgi body. See, it's a membrane organelle, Golgi body, Golgi apparatus. Say 0.4. That's the so because there are many things. There are many membranes inside the cell. If it has a like a transmembrane region, does it mean it is on the or on the cell wall or near the cell wall, or is it inside the cell on a endoplasmic reticulum or Golgi apparatus? The cell is full of membranes. Okay, that's why this membrane thing is very important. That's why we have all these search engines to understand, the function where it is in the cell. So results suggest that this protein is a plasma membrane bound protein, yet it is probable to find it on other cell surfaces with lower chances. So lower values for Golgi apparatus, and higher chance for plasma membrane. The 0.6. See, for 0.3 for endoplasmic reticulum, and then 0.1 for lumen, endoplasmic reticulum lumen. Where are you? I mean, so it's just, lots of spaces, for life to exist. And CBI conserved domain search. So again, you see all these domains. You see the domain, and then see all these domains. That is the query. That is the length of your protein, and that's just that's the cytochrome C domain. That's the values. Energy production and conversion. Cytochrome C. The green region. CCCA. See this one. It is important with a lower E value. Let's continue. Conserved domain architecture retrieval tool. So let's look at similar domain architectures. That's the whole sequence. Cytochrome C domain, PQQ domain, PQQDH, the hydrogenases with the pyrroloquinone, quinone as cofactor, like ethanol, methanol, and membrane-bound glucose dehydrogenases. The alignment model contains an eight-bladed beta propeller. Okay, so let's continue. My final blast for the queries sequence was NCBI CD conserved domain database. First hit protein displayed a central long domain of PQQ dehydrogenase with a cytochrome C domain at the end. Page up. So this was kind of a conserved domain. Like it's just so kind of a final, structure of the protein, kind of final architecture, combining everything. So now this is the 3D structure. You can generate this on the website. Again, you see a calcium element here, there's a calcium here, say calcium binding, regions. You see that's the molecule, PQQ molecule. So that's the molecule, and binding here. See in the binding in the center. Remember the domain, the PQQ domain was located in the center. And remember the helices, and then so these are the beta sheets, and then the helices are the kind of and then the central part. So that's the molecule. That's the PQQ molecule. That's the structure. The molecules are smaller, as you see. Remember, this thing is huge, 1100 amino acids, and this is just one molecule, sitting in the center. And all these loops, and see all these loops. This is like needle in a haystack. That is what biologists do. Searching for needles in a haystack. I mean, it's just that's your needle. It's just the time consuming, the nerve wracking, It's not easy to just to make sense out of this mess. Uh, Overall summary: my annotation for given protein is that this is a bacterial pyrroline quinoline quinone dehydrogenase alcohol dehydrogenase with pyroquinoline quinone as a cofactor. So it's a dehydrogenase with that molecule as a cofactor. It's an enzyme. This alignment model, this alignment models display several beta propellers on the N-terminal. The prosthetic PKK group, PQQ group, is located in the center of the domain. See, as I shown earlier. Page up. See, it's the center. That's the center of this is the 3D structure. You can generate it on the website as well. By looking at the data. There are generators. 3D structure generators built in the website. You can use. The prosthetic group is located in the center of the domain and is bound to a calcium ion. See, drink your mineral water. Calcium is good for us. This enzyme for bacteria too, and then remember there's gut bacteria too. This enzyme is an oxidoreductase, which can reduce substrates like ethanol, methanol, and glucose in some organisms. It's a detox enzyme. It's just basically that's what it is. It's just and then if you generate some alcohol in your gut, and then the bacteria use these enzymes to detox it or to process it. So it can reduce substrates like ethanol, methanol, and glucose because glucose is no different than ethanol and methanol. People don't want to understand it, but just check the molecular structure. Stuff is full of hydroxyl groups. Okay, I mean it is what it is. This enzyme might This enzyme might also utilize cytochrome C as a cofactor for other reduction reactions. That's the domain, cytochrome C domain. For other reductions, since it has a cytochrome C domain on the C terminal. So maybe it carries out a different reduction reaction other than reduction of ethanol, methanol, and glucose. It has a different function as well with this domain. I conclude that Interpro entry 11047 is the matching known protein for this query, which is quinoprotein alcohol dehydrogenase. This protein is predicted to have a transmembrane and terminal part confirmed by Interpro and MSET prediction. Okay, so and that is my drawing based on the the results. This was the homework, and then and maybe it wasn't. See, maybe it was. It is localized in the periplasmic space, and in this case, I should have mentioned, periplasm in the figure. This is my drawing, like see helices 13 to 30. See, this is the inside first terminal region. That's one amino acid one. See, I said inner outer. I and O, and then see that the total I have listed 6 helices, their length, and then see that's the outside loop, 145 to 315, and then this is the inner loop all the way to 1100, I should have mentioned periplasm here correctly. And maybe that is my and then remember, this was my actually first class on genomics, but I did my best, as you see. So this is the localization of the search protein, that professor gave us as a query model, and then that's how I did. This is the end of the talk, ladies and gentlemen. Thank you very much for listening. Please subscribe to the channel. I love you all. And this is again for high school students. This is AP biology level education. Okay, so it could be challenging. I don't know, but we are living in the space age. Okay. I know life is getting harder and harder. There's lots of data out there, but eventually, what can you do? The stuff is piling up. This information is piling up, and then we have to use it. All these proteins, and we can use these proteins, these enzymes, for reducing environmental pollution. Like, remember, this is a detox enzyme. It breaks down ethanol, methanol, and it works on those molecules. It's a dehydrogenase. The same thing for mushrooms. The mushrooms are organisms which have strong alcohol metabolism. They are very, the lignin, the stuff in the woods, lignin, the material is kind of an alcohol. It's an alcoholic substance, and it's important to understand these enzymes. And mushroom fungi has many enzymes to degrade these alcoholic substrates, including lignin and lignocellulose and other things. And so these enzymes have a job, have a function to do, and they have a localization. And either they are secreted as an enzyme. See, this protein is located on the membrane. It doesn't exist in a liquid state. It is not secreted. It is localized on the cell membrane. They all have different functions and different localizations. And oftentimes the localization tells you about the function. Every organism or enzyme or protein has a space, has a location where it can function. Okay, so spaces, locations are important. Without those, you cannot function. Okay, so this is the take-home message. It's not all about what you are, who you are. You need a place to function. You need a place to function. Without this place, you are useless. Thank you very much. Please subscribe to the channel.